So our first speaker is Joan Steitz, who is the professor of molecular and biophysics and biochemistry at Yale University. She's had many awards in her career, most recently the Lasker Award in 2018. And she was a postdoc in cell biology here at the LMB from 1967 to 1970. And she's very famous for all of her work on RNA, etc. And she is going to give us a talk with no slides.
Okay, so you just heard that I was at the LMB between 1967, late 1967 and late 1970. And I thought what I'd do is sort of give you a flavor of what things were like here then and what they were like particularly for me. So the reason I came here had not to do with me, but to do with my husband. So I was a graduate student with Jim Watson at Harvard, where I had studied the RNA phage, figured out some things about one of its genes. And I'd gotten married by the time it was time to decide what to do next. And my husband was a protein crystallographer, so there was no other choice in the entire world but to come to the Mecca. And this was the Mecca. And so this was where we were coming, regardless of what I might do. So Jim Watson wrote to his friend Francis. I don't know exactly what he said, but he must have said something relatively nice because I found myself accepted as a postdoc in the cell biology division. When we arrived here, I of course went to talk to Francis about what I might be doing. And he said, well, you know, we really don't have a project and we really don't have a mentor for you. So I thought maybe you could go do something in the library. And I knew that theoretical work was not my forte, in contrast to experimental work. And what I did was to go around and talk to all the people in the cell biology division. Now that constituted primarily American postdocs, all male, except for me. There were about a dozen of them who were working on various aspects of tRNA and I don't know, can't remember, some on protein synthesis. And I went around and talked to them and tried to get suggestions as to what they thought I might do. And I found that several people mentioned to me a very, very interesting project, very challenging project, which was the idea of taking ribosomes, binding it to a particular messenger RNA, getting the fragments of the ribosomes that were fragments of the messenger RNA that were protected by ribosome binding under conditions of the initiation, not the elongation of protein synthesis. And then using the new methodology that Fred Sanger was working out on the second floor, we were on the third floor, is that correct? We were on the third floor. We were on the second floor. Okay. Fred was on the third floor. Anyhow, that he was working out to sequence RNA, and because nobody knew this was, everybody was studying bacterial messages and their phages back in that day. Nobody knew where ribosomes started on a messenger RNA. Did they thread on from the 5' end, as we now know they do in eukaryotes, or did they just find the position somehow, some other way? And none of them dared take this project on because at that time, a typical postdoc was 2 years, and they were all afraid that it would be so challenging that they would have no results in 2 years to be able to go back and give a job seminar in the States in order to get an assistant professor position. Now, during my entire training, I had never seen a woman science professor. I'd never seen a woman head of lab. So I never expected to be in a position of competing for a faculty position. And I thought I would be a research associate in somebody's lab after my PhD. And I decided, why not take on this project? This sounds challenging and exciting. You know, maybe I'll make it work. And what then happened was Mark Brecher was kind enough to give me a little bit of bench space. We discussed it and decided it was about as long as this table or maybe a little bit shorter. And also lent his expertise. Now, I had one advantage. I knew how to make P32 labeled phage RNA. We knew it at that point. We thought it had 3 genes on it. And what Mark knew was a lot about the initiation of protein synthesis, which was being worked on by several people here. And so he knew the conditions that would be needed to make these complexes. The challenge then was to trim with ribonuclease and somehow fractionate the pieces protected by the ribosome from all the rest of the junk and get those pieces and do the Sanger methodology to sequence them. And that turned out to be the stumbling block. I spent about a year using sizing columns to try to fractionate the bigger bound stuff from the little pieces. And it's not a good idea because it got diluted. The RNA stuck to everything. I'd set up the fraction collector and come back the next morning and find my experiment that the fraction collector had stuck in the middle of the night. My experiment was all over the place on the floor. And I was about to give it up after a year because I just couldn't get the first step. And I remember having a talk with Sidney Brenner at that point. And Sidney said, you know, experiments can sometimes be like a bad marriage. You have to give them a second good try before you give them up for good. And I can't remember what happened, but for some reason, I had thought about using sucrose gradients, which everybody would do to fractionate the ribosome bound stuff from the other stuff. And I did that and it worked. And from there on, I got help from Bart Burrell and Fred Sanger's lab and a lot of other people on the sequencing. And it went quite fast to actually see the sequences of 3 sites bound by the ribosomes and R17 phage RNA. And that got me a Nature paper. So by late it was the spring of 1970 when we were scheduled to return after 2.5 years or so. I went with my husband back to Berkeley, where he had set up a job before we'd even, being a protein crystallographer, he had set up a job even before we had left to go to the LMB. And we went there and he had to teach some course or something, and we were there for the spring and several months. And when he inquired, I'm embarrassed it wasn't me, but when he inquired whether there might not also be a position for me somewhere at Berkeley, he was told, oh, but all our wives are research associates in our labs, and they love doing that. And with that, Tom and I had both given talks at various places on our way to Berkeley, and several of them had offered us both assistant professor positions. And we chose Yale, which turned out to be a very, very wise choice. Fred Richards, who was a protein chemist, but also a very, very broad thinker, realized that a powerful combination of structural biology and all its flavors and molecular genetics would be a wonderful way to build a new department. And he had just been given that department. I speculate, I think it may be true that Sidney Brenner had advised the then Yale president, Kingman Brewster, that this would be a good thing to do at Yale. And so we joined that department and have been there absolutely ever since. 52 years now for me. So I'd like to tell you about a couple other things that ended up happening in Cambridge, not directly related to research, but related. One day, Mark Brechter came along to me and he said, how would you like to be part of an experiment? And I'm an experimentalist, so I said yes. And it turned out Mark was a research fellow at Keys College. And he said, I'd like to put you up for member of the room. And he said, I will make the argument to them that this would be good for Keys in 2 ways. First of all, they could take precedence in terms of appointing a woman member of the room. Of course, there were no women who were had dining rights, being members of rooms at any of the Cambridge colleges then. But I will remind them that you're here temporarily. And you will be leaving, so this will not be a long-term problem. And so indeed, I got elected to being the first member of the room, first female member of the room at Caius College, which was very, very flattering. At the time, a man named Joseph Needham, who was a sort of biochemist and embryologist, was the master of the college. So we'd often sit next to him at high table. Stephen Hawking was also a member of the college at that point. He could still, with crutches, get up the few stairs to get to the dining hall. So it was actually quite interesting, but it used to take me at least a day or two to get up my courage to call the head butler and say, I would like to come in to dine. But once I got there, it was quite fun. They had nice sherry hours. Was it afterwards? Forget. Yeah, they were. That was really quite an experience. The other thing associated with that I wanted to mention was that my husband was a tennis player, and there were some beautiful grass tennis courts owned by Caius College. And my husband would go along and say, I'm Dr. Steitz, a member of the room. And they would shuffle over and check their lists, and they'd say, welcome, Dr. Steitz. So that was great. So things did work out very well for us. In the end, we ended up going to Yale in the fall of 1970, as I said before, I think. And what I want to sort of add at the end is what else LMB meant. LMB meant meeting a number of people, our peers, who now live and do science or have done science in various parts of the world, with whom we became fast friends, and with whom we have enjoyed many occasions, vacations, visits, etc., over the years. And really an incredible group of people that was here at that time that we have continued to maintain friendships with. So that's been most valuable aspect of life. Obviously, I found a project that, as it turns out, set the basis for my career. Once we figured out a little bit later, it wasn't immediately obvious that what those sequences were, is they had the ability to base pair with the 3' end of the small ribosomal RNA, and that was how the sites were chosen. The idea that base pairing between 2 RNA molecules might be something that would also be important in other aspects of gene expression turned out to be a gold mine, because we found many, many others, including the spliceosome, where, of course, it made sense that in the components of the spliceosome, if you have RNA molecules, you should be looking for complementarities in the substrates, and that's exactly what happened over the years. And finally, I do want to say that one of the things that I've gotten the most pleasure out of is teaching and mentoring students. In particular, it turns out, undergraduates. And the Yale undergraduates have been fabulous to work with over the years. And I discovered that it's almost as much fun to share the joy of discovery with a younger, talented colleague as it is to make that discovery yourself. And so I can't say anything but thank you very much to the LMB and all the richness it provided, both for science and for life. So thank you.
Thank you for a fabulous talk. Most of us are absolutely terrified if they haven't got a slide at the front.